Well, thanks very much, first of all, to the organisers of OHBM for inviting me to do a talk. It's a real privilege. I must admit, when I was first asked, I wasn't expecting to be doing it under these situations, these circumstances, in my own house at the end of May. But it's a real exciting opportunity to be part of the first virtual OHBM. So let's just hope my 5-year-old isn't wandering halfway through. That's what I'm going to say. Fingers crossed. Okay, so I'm going to talk to you about the mapping of fast transient brain network dynamics. And this is, like everybody else who works in this area, built upon a huge body of work that's been done in analysing large-scale brain networks using modalities like fMRI. And here's just one example by one of my colleagues in Oxford, Stephen Smith, who took the strength of connectivity from functional connectomes and linked it to behavioral traits in the human connectome project data. And this is just one example of the ever-increasing quality of information we can extract about large-scale brain networks, and the spatial detail is becoming ever more impressive. But I think it's fair to say that this is typically from a somewhat static perspective, and that is we typically tend to time average over the course of an entire scanning session in order to obtain our brain network estimates. And clearly that's provably been very useful in the field, but I think it's fair to say the brain is a highly stochastic and dynamic system. And so what we would argue is that what's underlying these time average brain networks is some idea of what we might call transient brain networks that are inherently coming and going over time. And it's only when we average the data over the course of a scanning session do we get the somewhat different perspective that the time average brain network is the main entity. So the work that I'll be talking about today is techniques for identifying and inferring these underlying transient brain networks, with a particular emphasis on extracting the dynamics of those networks and how they switch over time, looking to link them to task, behavior, and disease as well. So one way that you might go about doing that is of course using sliding window approaches, whereby we look to compute our functional brain networks within limited time windows. And then we can slide that window through time, building up a time course of our functional brain networks. And this has been proven to be very useful, but there is one sort of basic limitation with the approach. And that is that you have to have a window that's wide enough in order to be able to infer the functional brain networks accurately, and not just end up with what some people would refer to as spurious fluctuations in the functional brain network. So even in modalities like MEG, where we have exquisite temporal resolution on the millisecond timescale, people tend to use sliding windows on the order of several seconds in width. So this really is a real shame, because we're missing out on the very fast dynamic information at the kind of timescales you'd associate with the fastest cognition, so 10, 100 millisecond timescales. So we looked to come up with techniques that could circumvent this limitation. And the main technique I'm talking about today is a technique of hidden Markov modeling. And one way of thinking about this is kind of like breaking up the sliding window into a bunch of potentially much shorter lived visits to some idea of a repeating state. And the activity that you record when you come into that state can be characterized by a distinct functional brain network. Now, a crucial idea is that if you were to try and estimate that functional brain network from an individual visit, there would be far too little data. You just couldn't do it. However, if you can identify when you come back to the same state at other points in time, then you can pool over all of those different visits to get a really good estimate, a really accurate estimate of your functional brain network. So that might be state one. And then, of course, at other points in time, the brain visits other states where each one is characterized by its own distinct functional brain network. And as I say, this description of the brain activity is a sequence of states where each state is characterized by different networks, is what we can infer with the technique of hidden Markov modeling. So this is the generative model for the hidden Markov model. At the core of it are the parametric descriptions of the state time courses that tell you about which state you're in at each point in time. So these are, of course, binary state time courses, and we also assume that they are mutually exclusive. So you can only be in one state at any one particular point in time. We then have what we call the observation model that says if you know what state you're in at a particular time point, then we can make a prediction about the new imaging data that we're going to record at that time point, be it fMRI or MEG or EEG. And so for this, there's a whole swathe of different observation models we could look to use, but typically the one that we use is a multivariate normal distribution. And this it provides different descriptions of the nature of the functional brain network that's active when a state switches on. So for example, we can extract information about the amount of activity that's occurring in each of the brain regions. But we can also look at the covariance matrix that's at the center of the multivariate normal distribution and look at the off-diagonal correlation information to tell us about functional connectivity, using obviously correlation as a metric of functional connectivity. And crucially, we have a different multivariate normal distribution and therefore a different functional brain network description for each of our states. So we infer on this model probabilistically using variational Bayes techniques, and that allows us to simultaneously infer the state time courses and the observation models for the different states. And it essentially iterates back and forth between them to make sure that they are consistent with each other. And it's worth pointing out that whilst we do assume mutual exclusivity in the state time courses, we are doing this inference probabilistically. So there is a soft assignment to the state time courses in terms of the inferred states. So this is a very flexible approach, essentially an unsupervised learning approach that can go into different data sets and identify these switching states. And so it can be readily applied to different modalities in a similar way to ICA. I'm not going to talk about applications in intracranial data today. I'm just going to talk about non-invasive human data. And I'm going to start with an application to fMRI. So this is work done by Diego Vadore, who's one of the main people who's worked on the HMM method and developed the toolbox that we use in our applications. And in this first application to fMRI data, the initial ambition wasn't particularly lofty. It was simply to try and see if we could infer sensible dynamic networks in resting fMRI data. And then secondary to that, if we were successful, we wanted to see if the nature of the dynamics could be related to behavior. So we used data from the human connectome project to do this, and we inferred 12 states. And it's worth just saying at this point that when we infer 12 states, and in general, when we say we're inferring a certain number of states, we always make sure that the results that we get aren't fundamentally critical on that exact choice of number of states. And so we check that the results we have do not fall off a cliff as we move up, say, to 13, 14, 15 states or down to 11, 10, 9 states. So we want to make sure the results generalize over the model order to a reasonable extent. And that was the case here and in all the studies that I'm sharing today. So here I'm showing you 6 of the 12 states that we inferred, just for brevity. And this is showing the relative activity map, so the amount of activity you have in different brain areas in the 6 states. And we see the same mix of primary sensory motor and cognitive networks that we typically see in resting state. So there's nothing particularly different here. There are some minor differences, but the main difference that we're interested in is in the dynamic description, for which we turn to the state time courses. So we can actually look at the state time courses. Here we binarized them following the inference. And then we can look to summarize those state time courses using different summary measures. For example, we can look at the proportion of time spent in a state over time, such as the fractional occupancy. We can look at the interval time between state visits, or we can look at the lifetime of a state, or the dwell time, as it's sometimes known. And that's what we're looking at here. We're looking at the distributions of the lifetimes, so the time spent in a state before moving into a new state for the 12 states. And we can see here that a typical lifetime is on the order of 5 or 6 seconds. And so we're seeing switching on timescales as fast as you could really expect to go, given the hemodynamic limitations of the fMRI data, the fact that we're looking at the brain through the blurred lens of the hemodynamic response. Okay, so we then look to see if the sequencing of the states has some kind of interesting temporal structure in it, or whether it was completely random. And to do this, we looked at the transition probability matrix, which is inferred as part of the hidden Markov model. Because we have 12 states, this is a 12 by 12 matrix telling you about the probability of moving from any one state into any other state. And you can see just by eye, there's already evidence of like a block structure to this matrix. And indeed, if we submit the matrix to a modularity analysis, then we find that there are two significant modules that pop out. We call these metastates, and metastate 1 contains 5 states, and metastate 2 contains 7 of the states. So what we can then do is look at the observation models for these states that make up the metastates to see what they correspond to in terms of the spatial networks. And here I'm showing that information aggregated over the different states that make up a metastate using the functional connectivity summary measure of connectedness, which relates to degree. So you can see in green and blue that metastate 1 corresponds to primary sensory motor regions, typically, whereas the metastate 2, shown in yellow and red, corresponds to cognitive networks, such as the default mode. And so what this is essentially telling us is there's a tendency for the brain to cycle either between cognitive networks or to cycle between primary sensory motor networks with a much lower probability of switching between them. And of course, that does happen. And indeed, if you look at individual subjects, they do mix between the two metastates. But you can observe a large amount of between subject variability in the amount of time they spend in particular metastates. And so we reasoned that this might be like a signature of an individual's behavioral traits, and look to relate the amount of time spent in metastates to the behavioral trait data information in the human connectome project data. And this is what we found. We essentially found that people who tend to spend more time in the cognitive metastate are more likely to be associated with positive behavioral traits, whereas people who spend more time in the primary sensory motor metastate are more likely to be associated with negative behavioral traits. And then perhaps unsurprisingly, given the trait link, we also found that the amount of time spent in the metastate was also heritable using the twin structure in the HCP data. So this is a really good example of the kind of information we can extract, not about the strength of connectivity as you would do in a static time average large-scale brain network, but using the dynamic information from this transient perspective we get from the hidden Markov model. So as a summary of this first part, we've seen how the HMM provides descriptions of network state dynamics. The sequencing of the states that we found in resting fMRI was not random. Instead, the brain tends to switch between either primary sensory motor states or cognitive network states. And the time spent in these two metastates was predictive of behavioral traits using the human connectome project data. Okay, so that first part was an fMRI. I'm now going to switch to the modality of MEG. And I first got involved in working on MEG, having been an fMRI analysis person before that in about 2010, 2011. And it was a particularly exciting time in MEG, at least I thought so, in as much that people were looking to take the ideas of how you infer functional connectomes from fMRI data and look at how we could do the same kind of things in MEG. And it's a particularly exciting proposition because the amount of temporal detail you can extract from the MEG is potentially much higher, albeit at the slight expense of spatial detail. So one of the challenges that we had to overcome though at this time was the issue that you're now exposed to the fact there can be communication delays between brain areas, which is soaked up by the hemodynamic blurring of the fMRI. So we now have to deal with this issue. So we're essentially saying that if we look at the underlying oscillatory activity in different brain regions, because of the delay in communication, the oscillatory activity can be lagged in terms of the oscillatory phase from region 1 to region 2. And so we can't just use raw correlation anymore as a functional connectivity metric. It would just give us 0 in this case because they're 90 degrees out of phase with each other. So we turn to other techniques. So for example, you can use phase locking or coherence measures that tell you about the extent to which the oscillations are synchronized at different lags. But what was found to be the most robust way of dealing with this issue was not to work with the raw time courses, but instead to work with the amplitude time courses, which you can compute using things like a Hilbert or a wavelet transform. And these are essentially at slower timescales and capture the envelope of the oscillations and therefore soak up this communication delay issue, thereby allowing you just to use simple correlation between brain regions. And so when people did this, they found that they could find significant functional connectivity between brain regions that you expect to be functionally connected. One of the new insights they got from this, though, was that the oscillatory frequencies at which the amplitude correlations was found upon was in the low frequency range, so between 4 and 30 hertz typically. So we got involved with a piece of work by Matt Brooks at Nottingham University in the UK at around this time. And he was looking to try and get ICA techniques working in MEG in a similar way to fMRI in resting state data in order to find resting state networks in MEG. And this hadn't really been done particularly convincingly before in the same way that showed good correspondence with fMRI data. So we took the tricks learned from the sort of seed-based correlation approaches. He worked with the amplitude time courses, these blue time courses, in this 4 to 30 hertz range, and fed those into ICA. And when he did this, the RSNs that we're used to seeing in fMRI popped out very nicely, showing pretty good correspondence between modalities, albeit with less spatial resolution in MEG, of course. One of the downsides of the analysis, though, was that in order for it to be robust and to give us good SNR in the amplitude time courses for the ICA to work on, Matt needed to use a small amount of low-pass filtering such that he smoothed the data to the tune of about a second or two. And this means that obviously it works very well, but the downside is we're missing out on the ability to access the information at the fast sub-second, 10, 100 millisecond timescales that we'd like to be able to use in the MEG data. So this is where we turn to the idea of hidden Markov model. And our kind of interest in this was actually also inspired by some work by Gustavo Deco at the time, who was working on biophysical models to predict oscillatory activity. And he found there was a tendency for these biophysical models to predict transient bursting oscillatory activity. And other people were showing similar things as well. And so we reasoned that the HMM would be a really good way of capturing these transient bursting oscillatory kind of events in the network behavior. So we took a similar analysis as the ICA approach. We worked with data in this low frequency range between 4 and 30 hertz, and we computed amplitude time courses. And then we looked to train the HMM to infer state time courses and the state-specific functional brain networks. And this is what we found. I'm showing you 4 of the 8 states, first of all, in terms of the amount of activity that you see in each of the 4 states shown here. And we see the same mix of primary sensory motor and cognitive networks that we're used to seeing in resting state. But if we look at the state dynamics, this shows really very fast switching as we hoped it would. Indeed, if you look at the first 10 seconds of the first subject, you can see that there's a switching between all of the 8 states here. So if you were to use the sliding window approach, you would completely smear together all of these state visits and you'd lose the temporal detail that we can get with the HMM. We can look at the speed of the switching by looking at these lifetime distributions again, as we saw in the fMRI data, but here the switching is typically on the order of 100 milliseconds. So we're seeing these resting state networks switching on timescales orders of magnitude faster than have been seen previously using other approaches. So we can, of course, summarize the state time courses using other metrics, for example, the fractional occupancy or the interval time between state visits, and look to link those to task, behavior, and disease. And this has been done in a few studies since. I'm just going to highlight one example of where it's been linked to disease. This is a really nice study done by David Salat's group at NGH, where they ran the HMM on resting state MEG data in an Alzheimer's study. First of all, they found very nice replication of the spatial map information that we saw in the original HMM application to MEG data. But then they had a look at the dynamic summary measures and looked to link those to disease. And what they found was that the fractional occupancy or proportion of time spent in particular states was predictive of disease, such that people with AD tend to spend less time in the default mode network states and the dorsal attention network states in the resting state. So this is a nice example of the kind of way in which we can use the dynamic descriptions of the connectomes or the functional brain networks to link to disease. So one of the issues that we were kind of challenged with when we first started using the HMM on MEG data in particular was this speed of switching. So we had this 100 millisecond timescale where the states were switching very quickly, and people quite rightly questioned if the fluctuations were meaningful or are they just spurious and caused by fitting to noise. So obviously we can do various things to kind of test that, but one of the most compelling things to do is to run it on task data and see if the fast fluctuations are linked to task at these fast timescales. So this is work done by Andrew Quinn in Oxford. He worked with a very simple faces versus scramble faces task MEG data set. And what we did is simply fit the HMM in exactly the same way that we fit the HMM to resting state data. So we have no knowledge of the task timings. It's completely unsupervised. This gives us the state time courses as well as the functional brain network descriptions. What we can then do is bring in the task timing information at that point. So the timing of the presentation of the faces. And then we can do an evoked response analysis time locked to those images. And these evoked responses that we get for each of the state time courses can be thought of as the probability of being in a particular state time locked to the presentation of the stimulus. And what we see when we do this, and this is generalized across a number of task studies, is that the state time courses show strong task dependencies at these fast timescales, sub-second, 100 millisecond timescales. So state 1, for example, which corresponds to primary visual cortex, shows an increase in occurrence at about 100 milliseconds post stimulus presentation, consistent with a visual evoked response. And so, seeing these fast fluctuations linking to task in a meaningful way gives us confidence that generally, it's not definitive, but it gives us confidence that generally the fluctuations we see elsewhere in the resting state are also potentially meaningful. Okay, so we have arguably these meaningful fluctuations at 100 millisecond timescales in the resting state's MEG data. And at the same time, we of course have fluctuations in fMRI data at much slower timescales, sort of 10-second timescale. And so an obvious question is, what links these two different timescales? Is there a link between them? Or maybe they're completely independent processes. So one obvious way in which they're linked is that these HMM state time courses that we're seeing not only contain this really fast switching, but they also contain a long-range temporal structure, whereby if you look at slower timescales, there's kind of a temporal structure, a clustering of the information. And we saw a hint of this already in the fMRI analysis, where we saw the sequencing of metastates. So what we're basically saying is, one way of testing this is if we take the MEG state time courses and we convolve them with the low-pass filtering effects of a hemodynamic response function, do we have meaningful resting state fMRI fluctuations that are left over? Now, we can't explicitly test this with fMRI and MEG because we can't get simultaneous fMRI and MEG data. But we can, of course, get simultaneous EEG and fMRI data. And this is what Tatiana Sitnikova has done recently in a study where they acquired resting EEG and fMRI concurrently. They had 128 channel EEG, and using that kind of higher density EEG, they were able to very nicely replicate the HMM on the MEG data that we've seen previously, given the same mix of spatial maps, broadly speaking, and the same speed of dynamics. They then took the HMM that had been inferred purely on the EEG data and took the state time courses and HRF convolved them and then correlated them with the fMRI data. And this allowed very nice looking resting state networks to pop out in the fMRI data. So for example, here's the DMN inferred in the EEG using the HMM, and then that's HRF convolved and then correlated with the fMRI. And here you can see the corresponding DMN in the fMRI data. So this is a nice demonstration that this link does exist between the EEG and the fMRI fluctuations. Caused quite clearly, and it's the only way it really could be caused, by the presence of long-range temporal structure in these EEG state time courses, and by implication in the MEG state time courses as well. And we can go directly looking for this long-range temporal structure by using measures on the state time courses themselves on the electrophysiology. So measures like the Fano factor, for example, and that allows us to capture the extent to which there's temporal clustering of the visits to state. So if you like, the extent to which there's bursting in the state visit behavior over time. And what we see is that there is, for all of the states that we inferred in this decomposition here, we can see that there are indeed evidence of burstiness in the Fano factor at the kind of fMRI timescales. Okay, so as a summary of this part of the talk, we've seen that the HMM on resting states, MEG and EEG data reveals transient RSNs on 100 millisecond timescales. And the rate of occurrence of RSN states is predictive of disease and of tasks. And the HRF convolved EEG state time courses correlate very nicely with fMRI RSNs, we think thanks to the long-range temporal structure that we see in the EEG state time courses alongside the fast switching information. Okay, so everything we've seen so far has been using the HMM on on amplitude time courses. So this is the envelope of the oscillations that we talked about earlier, which we work with in order to deal with this communication delay between different brain regions. And that's worked really well, as we've seen. But the question is, can we do better than this? And the reason why we might think that we can do better is the fact that the amplitude time courses are inherently at slower timescales than the oscillatory activity. And so we're potentially missing out on faster dynamics than we get with the amplitude approach. Furthermore, by not being able to work with the underlying raw oscillatory activity, we could be missing out on information about these specific or more specific mechanisms of interaction between brain regions. So for example, measures of synchronization and phase locking. And that's particularly important because synchronization of oscillations has been put forward as one of the mechanisms by which brain areas basically integrate information over large swathes of the cortex. Okay, so what we can do is think about other ways we might access this information and work with the raw time courses. And the solution that we've come up with is a technique called time delay embedded HMM. And we actually took this idea of time delay embedding from the world of generalized synchronization that we were playing around with at the time. And time delay embedding is essentially quite a simple idea. All you do is take the oscillatory activity from a particular brain region and then include alongside that time delayed or lagged versions of that time course. And you do that for different lags. So that's for brain region 1. And then of course, we repeat the same process for all brain regions, including their own delayed and lagged versions of those time courses. And what this allows the HMM to do is come in and find where there are correlations between brain regions at different lags. So for example, in this cartoon example here, we can see that brain region 1, a lag of 2 tau, is correlated with brain region 2 at 0 lag. Okay, so that's very nice. It allows us to deal with the fact there's communication delays between brain regions. But in fact, this approach kind of gives us more broader information. If we look at a more general setting, here we have, again, 2 brain regions, and we're talking about 2 hypothetical states here. And so what the HMM is going to do, we're still using the multivariate normal observation model with this approach. And so we're using the covariances of those multivariate normal distributions to work out what are distinct states. But now, of course, the covariance is being calculated in this time delay embedded space. It's telling you about correlations both between brain regions, but also between lags within brain region and across brain regions. And in so doing, it can capture rich information about synchronization, for example, or indeed spectral information in individual brain areas. So in this example here, we have in state 1, this red state, synchronization between brain region 1 and brain region 2, and that gives a very distinct pattern in the covariance matrix in the time delay embedded space. Whereas state 2 has no synchronization within the same frequency band and shows a very different pattern of covariance in the time delay embedded space. And if you look at the information we can get about the covariances and translate it into the Fourier domain, these covariance matrices can be thought of as telling us about state-specific spectral information in different brain regions, but also, most interestingly, from our point of view, state-specific maps of synchronization of networks in networks, capturing the synchronization between different brain regions via the time delay embedding. So we look to use this approach and apply on resting MEG data to see if we could find evidence of brain-wide networks of synchronization. And as I mentioned, we're particularly interested because the idea of synchronization has been put forward as a fundamental mechanism of communication in the brain in terms of how brain areas integrate over large distances. I think it's fair to say that before this piece of work, to some extent, it'd been quite hard to obtain very strong, believable phase locking networks or synchronization networks in resting state data. And we reasoned that the reason why they weren't looking quite as compelling as we might hope is that people were looking at too slow timescales because of the issue of averaging over time using, for example, sliding window approaches. But we hope that by using the HMM, then we could access much faster timescales and reveal these networks of synchronization bursting on these fast timescales and allow us to see them more strongly because we're not smearing them out by averaging over time. So the first thing we can look at is the state time courses that we get from using the time delay embedded HMM. what we can see now is that the lifetime of state visits is on the order of about 50 milliseconds. So about twice as fast as we saw with the amplitude HMM. The first thing we're looking at in terms of the functional brain network descriptions is the spatial maps, telling you about the amount of power that you have in each brain area between 1 and 30 hertz. And we're showing you it here for 4 of the states of the 12 that we inferred. And we see the same mix of primary sensory motor and cognitive networks that we see typically in the resting state. However, there is one slight difference here in that the anterior and posterior DMN represent a kind of splitting of the DMN putatively into 2 different networks. And we'll come back to that in a moment. But the first question that we wanted to answer was, are there more compelling networks of synchronization? And we think the answer is yes. By looking at these fast timescales, we reveal stronger networks of synchronization between the brain areas you'd expect in these particular networks. So for example, in the anterior default mode network, we see strong phase locking synchronization between anterior regions of temporal lobe and frontal cortex and medial prefrontal cortex, and also connectivity going backwards to the PCC. Whereas by contrast, when we look at the posterior default mode network, we see the strong synchronization network in the more posterior regions, as the name might suggest, but between areas of parietal cortex, precuneus, and PCC. Okay, so we can actually drill down a bit further, have a look at these networks of synchronization in more spectral detail. And when we do that, we can see, for example, with the anterior DMN, that the frontal network of synchronization is happening predominantly in the delta and theta band, with nothing happening in the alpha band. By contrast, if we look at the posterior default mode network, it's doing most of its work in terms of synchronization in the alpha band, particularly in this posterior set of regions that we saw earlier. There is some connectivity in delta theta as well, but it's much less, and it's more frontal. And this relationship between anterior and posterior DMN and oscillations has been shown to some extent before. For example, in concurrent fMRI EEG studies by Dante Mantini back in 2007, they found evidence of an anterior DMN, which they referred to as a self-referential network, that was more strongly linked to delta and theta. And then they also found a posterior DMN, or sort of main DMN as they refer to it, that was more strongly linked to alpha and to some extent beta as well. So we're capturing the same information, broadly speaking, here with this HMM analysis, but with the additional perspective that we're now providing evidence of there being strong synchronization between the brain areas, these networks of phase locking or phase coupling. And also that these networks are arguably switching on these fast 50, 100 millisecond timescales. So as a summary of this part of the talk, we've seen that the time delay embedded HMM allows us to identify transient networks of synchronization at 50 to 100 millisecond timescales in resting MEG. Synchronizations occur in different frequency bands for different networks. For example, the anterior DMN synchronizes in the delta theta band, and the main posterior DMN more so in the alpha band. And we think this information is available because we're accessing these faster timescales with the HMM. Okay, so in the last segment of the talk, I'm going to focus on the use of the HMM in the context of replay. And this is, I'm sure people are very familiar with the idea of replay. Just briefly, it's been mostly studied in the animal literature, in rodents in particular, whereby when mice, for example, are moving through an environment, you see particular sequences of place cells firing as the mouse moves through particular locations. Of course, hippocampal place cells fire in relation to those locations. But if the mouse moves through the same route, then you'll see the same sequence of place cell activity. So that's pretty nice in its own right. But what's really compelling is that when you observe the recordings when the mouse is subsequently at rest, then you see the same sequence of place cell activity spontaneously replaying or playing itself out in this resting state. These replay events, reoccur at random points in time repeatedly, and they're also temporally compressed, such that the time between replay events, or the time between items within the replay of a sequence, is on the order of about 30 or 40 milliseconds. And they occur also during these high frequency oscillations that can be observed in the local field potential recordings, and so-called sharp wave ripples. these have been heavily studied in the animal literature, and they're thought to be associated with things like memory consolidation and augmentation and other related memory performance. Okay, so what's particularly interesting though for us is that recently it's been shown that you can measure replay of sequences in humans non-invasively using MEG. And this is work done by Zeb Kett-Nelson and more recently Yunzhe Liu as part of Ray Dolan and Tim Barron's groups. And what they did was not have people running around an environment. They instead had them sitting in the MEG scanner and observing or basically learning sequences of visual images. And the visual images, the sequences of visual images may or may not lead to rewards, providing the motivation for the sequence learning. And then subsequent to this learning, they're then subjected to a resting state MEG scan. And the idea is to find evidence of reactivation of the brain activity associated with these sequences of visual images. And so, of course, to be able to do that, previous to the behavioral learning and the resting state scan, they used a functional localizer in the MEG scanner, where people were presented with visual images that were going to be used in the sequence learning. And a brain decoder was trained in order to be able to predict what images were being represented in the brain activity given the MEG data. And then armed with this decoder, you can then use it on the resting state data to find when the particular visual images in particular sequences are being reactivated. And so when they did this, they found evidence of sequenceness, as they called it, evidence of replay of sequences. And this has now been replicated over a couple of different studies, and probably more that I'm not aware of. So this is very nice. And what we were particularly interested in is taking this ability to infer replay events in the resting state data, and then working with the same resting state data, look at the dynamics that we can see in the resting state networks using the HMM approach that I've been talking about today. And this can essentially allow us to link together the sort of field of replay that's been mostly covered in the animal literature with the field of RSNs that's obviously not exclusively, but been certainly studied quite extensively in the human literature. And they also allow us to link information that's typically studied in replay more spatially locally in hippocampal data to what's happening across the whole of cortex using MEG data and RSN network characteristics. And the reason why we can do this is because we have the ability to look at the same kind of time scale. So the replay events occur on these 30-40 millisecond time scale, whereas as we've seen, the HMM can identify network dynamics on the 50 or so millisecond time scale. So we have the ability to potentially do this. So as I say, we infer replay events and the HMM state time courses separately on the same resting state MEG data that's come from the replay experiment. And then we look to do the same evoked response analysis, whereby we basically epoch around the replay events, the different state time courses, and average over repeats of events. And this gives us these effectively these evoked responses of the state time courses that tell us about the probability of the state occurring time locked to the occurrence of the replay events. And when we do this, we see two states in particular that pop out. These correspond, first of all, to the main posterior DMN that we saw earlier, using the time-embedded HMM. I should say we're using the time-embedded HMM here as well. And then we also have a parietal alpha network that also pops out. Both these networks show predominant oscillatory activity in the alpha band as well. So these two networks are distinguished by being correlated with replay. They increase in occurrence in this 100 or 200 millisecond period around a replay event. But they also can be distinguished via some other phenomenon. For example, the extent to which they exhibit network synchronization. So here we're summarizing the amount of synchronization across the functional brain networks that we can infer for these two states. And they both stand out, particularly the posterior DMN. It shows a large amount of synchronization. You recall from earlier that we looked at the extent to which the state time courses show long-range temporal structure, which we quantified using the Fano factor. It tells us about the extent to which there's kind of temporal clustering or burstiness of state visits. And again, these two states particularly stand out from the crowd by having exceptionally high amounts of long-range temporal structure compared to the other states. And this is particularly notable because if you look at running the same failure factor calculation on the replay events themselves, they also show the same extent of bursting behavior at longer timescales as well. So clearly there's a link between the dynamics of these resting state network states and the replay events, both in terms of the fast switching, but also in terms of these long-range temporal dependencies as well. In the Enze Liu paper, one of the interesting findings that was quite tantalizing was that if you looked at the activity around the replay events without the HMM, they observed there was particularly heightened amounts of power in the high frequency range, so between 100 and 150 hertz. And this raised the possibility this could be linked to the sharp wave ripple events that occur in hippocampal recordings. And so we had looked to see if we had something similar happening in the HMM states, the resting state networks. And we found that in this case, it was the posterior DMN on its own that stood out from the crowd. So the parietal alpha is not looking exceptional in this regard, but the posterior DMN shows particularly heightened amounts of high frequency oscillatory power. And this is particularly interesting because the HMM itself was only inferred on the 1 to 48 hertz frequency range. So it didn't use this information when we trained the HMM. And if we look at the spatial distribution of the high frequency power associated with the DMN, we can see that it concentrates in the anterior temporal lobe regions, adding potential evidence to the idea that it may be linked to sharp wave ripple events in hippocampus. So how might we go about interpreting these results? We're talking about wakeful rest data here. We're not talking about sleep data. And so it seems reasonable to assume that particularly in wakeful rest, the brain needs to be able to organize replay events in such a way as to minimize the interference with ongoing cognition. And so that might lead to the idea that the parietal alpha network needs to switch on, as we see in the data, in order to inhibit bottom-up sensory processing during replay. And then at the same time, the posterior DMN switches on, providing brain-wide, very exceptionally high levels of synchronization across large swathes of cortex, supporting the integration of signals across the cortex, providing if you like, this is obviously slightly more speculative, but providing the ability for the event that's been kicked off in the hippocampus to propagate across the rest of cortex, to properly allow the replay event to occur on a cortex level. And with the link to high frequency oscillations, there's also the possibility that we have these synchronizations linking all the way up to the 150 hertz range. Okay, so I think regardless of these more specific speculative interpretations, this is new evidence that helps to broaden our interpretation of the functional role of the DMN when we bring together this replay work with the HMM and its ability to estimate RSNs at fast timescales. Okay, so just going to finish off by looking at some future directions for both the replay work and more broadly for dynamics of functional brain networks. So one of the interesting things that we found with the replay work was that these key features of the posterior DMN and the parietal alpha networks are that we can see them just in bog standard resting state data as well. In other words, the features such as exceptionally large network synchronization up to high frequency, and also the bursting on long timescales, this long range temporal structure. We can see both these things on normal resting state scans in the MEG data. So this raises the tantalizing possibility that we could look to try and use normal resting state data as a kind of surrogate measure of the quality and quantity of replay without having to go to the lengths of the intricacies of the replay experiment, so the functional localizer and so on. Now, of course, we can't do the same level of precise neuroscience with this approach, but it might provide a measure that can be used in clinical research, for example, that's much more readily accessible compared to full-blown replay experiments. As we've been using the HMM over the last few years, we've obviously learned its strengths and weaknesses. Clearly, no doubt, concentrating more on its strengths in today's talk, but there are a bunch of weaknesses that we've learned about over the years. One of these is the assumption of mutual exclusivity, so that only being in one state at one point in time. And the other is the fact that the HMM, through its Markovian assumption, only captures short-range temporal structure in its model. But that's not to say, as we saw, that the state time courses, when inferred on real data, can't capture long-range temporal structure. We saw that in the earlier analyses. And that's because the data, the information, the data override the prior constraint of the HMM's Markovian assumption, providing the long-range temporal structure despite the HMM rather than because of it. But nonetheless, that's a deficiency, and we'd like to use models that explicitly model long-range temporal structure and allow us to relax the assumption of mutual exclusivity. And so we're looking to techniques from deep learning, perhaps unsurprisingly, in the current climate of deep learning being quite a popular perspective on machine learning. And so there are approaches that are explicitly designed to capture long-range temporal structure, for example, recurrent neural networks and long short-term memory models, for example. And so the hope is by combining deep learning approaches with big data, there's a really exciting possibility in the future for the field in general of being able to extract ever more precise and more accurate information about the dynamics of brain networks using techniques like fMRI and MEG. So I think exciting times lie ahead. And I'll just finish by saying a big thank you to all the people involved in the work and to the Wellcome Trust as well for funding much of the work. And thanks very much for listening.